Hello, I'm Andrej Shevchenko. I'm group leader in Max Planck Institute of Molecular Cell Biology and Genetics in Dresden. Today, I am presenting our recent work on the lipidomics of non-alcoholic fatty liver disease, recently published in Journal of Lipid Research. In a nutshell, what is the non-alcoholic fatty liver disease? Normal patients have a healthy liver, but they can become obese, yet their liver still stays healthy. Then, by a poorly understood mechanism, their liver starts accumulating fat, and the number and size of lipid droplets increases, leading to a non-alcoholic fatty liver, or NAFLD. The progressive accumulation of fat leads to oversized balloon lipid droplets, inflammation, fibrosis, and develops into steatohepatitis, or NASH. If untreated, it leads to liver cirrhosis and cancer. Apparently, NAFLD is a lipid metabolism disease. However, if and how it affects the full liver lipidome is an open question. To analyze liver biopsies, we applied quantitative shotgun lipidomics. Basically, lipids are extracted from homogenized biopsies, total lipid extracts are directly infused into high-resolution mass spectrometer, raw spectra are processed by peak strainer, and lipids identified and quantified by Lipid Explorer software. Lipidome compositions, in which the abundance of each lipid is expressed in moles, is subjected to statistical analysis. Altogether, we analyzed 365 biopsies from patients from 4 disease groups and systematically quantified 316 lipids from 22 major classes, including membrane lipids, such as glycerophospholipids, cholesterol, phospholipids, and energy storage lipids, such as 3 and D-acylglycerols. As expected, the abundance of neutral lipids increased, but the bulk of membrane lipids did not change significantly. Once submitted to PCA, the lipidomes of steatotic and non-steatotic patients only differed by neutral lipids. Once 3 and D-acylglycerols were cleaned from the dataset, we observed no separation. The molecular composition of 3 and D-acylglycerols, such as the length and the unsaturation of their fatty acid moieties, also did not change. Basically, a steatotic liver accumulated the same type of fat that was compositionally similar to adipose fats. Common clinical markers did not serve as reliable classifiers of NAFLD, and therefore we asked if any lipid species can do any better work. Expectantly, the most specific classifiers were 3 and D-acylglycerols, with only marginal impact on fatty acid unsaturation. Interestingly, there were no membrane lipids on the list. However, with one remarkable exception of out-chain sphingomyelin species. However, we wondered if they are associated with some disease stage or can contribute to disease prognosis. To answer this and many other questions, we forged a collaboration with Josh Pauling in Technical University München. How the NAFLD story continues explains the next presentation given by Team Rosa.